Hello, welcome to World of Microbes. This is the second part of conventional diagnosis versus molecular diagnosis. Link of the first part of this video is in the description. In today's video we are going to talk about genotyping. Genotyping is the technology that detects small genetic differences that can lead to major changes in phenotype, including both physical differences that make us unique and pathological changes underlying disease. It has a vast range of uses across basic scientific research, medicine, and agriculture. Genotyping is the experiment that identifies the differences in DNA sequence among individuals or populations. It is used to understand the connection between genotype, the underlying genetic code, and phenotype, the observable organismal structure. Genotyping methods include DNA sequencing, analyzes all nucleotides in a DNA sequence. Mass spectrometry, analyzes the molecular mass of ionized particles. Molecular beacons, detect specific genetic sequences. SNP microarrays, detect single nucleotide polymorphisms. PCR-based methods, amplify specific DNA sequences. Genotyping can be performed on large-scale populations using high-throughput technologies like microarrays and next-generation sequencing. These methods would be discussed in detail in other videos. Restriction endonucleases are enzymes that recognize a specific DNA sequence, called a restriction site, and cleave the DNA within or adjacent to that site. They are also known as restriction enzymes or restrictases. Restriction endonucleases recognize specific DNA sequences, usually palindromes that are 5 or 6 base pairs long. They cut the DNA by splitting the bond between adjacent nucleotides. The cut can be flush, blunt, or have 5' or 3 overhangs. The number of fragments produced depends on the number of restriction sites in the DNA. Restriction endonucleases are used to insert foreign genes into vectors. They are used to characterize amplified DNA through restriction fragment length polymorphism, RFLP, analysis. They are used in DNA fingerprinting and forensic analyses. Restriction endonucleases are part of restriction modification, RM, systems. The RM system protects the host DNA from restriction. Restriction endonucleases are classified into 3 types, called 1, 2, or 3, based on their genetics and enzymology. Restriction endonucleases are available from many manufacturers of biotechnology products. When choosing a genetic marker, the primary considerations are the study's purpose, the level of polymorphism needed, the cost, and the availability of technology to analyze the marker. Factors like the organism being studied, desired level of resolution, and the need for high-throughput analysis will also influence the selection, with including morphological markers, biochemical markers, like isozymes, and DNA-based markers, such as SNPs, SSRs, RFLPs, AFLPs, each with their own advantages and limitations depending on the specific research question. Key points about choosing a genetic marker. Markers. These are visible traits like flower color or plant height, often used for initial screening due to their ease of observation but can be limited by environmental influences and may not be highly polymorphic. Biochemical markers. These analyze protein variations like isozymes, useful for studying population genetics but environmental factors and may not be as informative as DNA-based markers. DNA-based markers. Considered the most advanced option, offering high polymorphism and the ability to analyze specific genomic regions. Here are some factors to consider when choosing a marker. Polymorphism level. How much variation exists within the population being studied. Informative power. Ability to distinguish between different individuals or genotypes. Cost and accessibility, availability of technology and expertise to analyze the marker. Transfer. The marker can be used across different populations or species. Genomic coverage, how well the marker represents the entire genome. Applications. Personalized medicine, understand how genetic variants contribute to disease, drug responses, and therapeutic failure. Plant and animal breeding, identify and select plants with desired traits. Ancestry and disease tracing, trace the genetic origins of disease or ancestry. Inheritance and determine biological relationships. Pathogen typing and resistance screening. Identify pathogens and their resistance to drugs. Biodiversity monitoring. Track genetic diversity within populations. Do like and in the next parts of the video we will discuss the genotyping methods in detail separately. Do like and subscribe.